saying good afternoon and thank you for joining us for today's program, Saving the Florida Goldenaster, where we'll be learning about one of Florida's rare endemic plant species, Chrysopsis floridana, commonly known as Florida Goldenaster. So now on to our speaker. For today's program, we are joined by Cheryl L Peterson, who is the manager of the Rare Plant Conservation Program at Bok Tower Gardens. She has a bachelor's degree in botany and microbiology and a master's degree in cell and molecular biology. Her previous research involved biodiversity and genetics of marine invertebrates and studying the Florida endemic mint genus Conrandina. At Bok Tower Gardens, she works to establish new populations of rare species, including scrub lupine, Florida Goldenaster, Brooksville bellflower, clasping loria, and Savannah's mint onto protected lands. She has been recognized with numerous awards for her individual achievements and for the work of her rare plant conservation program. We are so pleased to have her with us today. Thank you for joining us, Cheryl.
Thank you, Stephanie. And so thank you, everybody, for your interest in the Rare Plant Conservation Program at Bok Tower Gardens. So even though I come representing Bok Tower Gardens, I'm not going to be talking at all about the gardens themselves. So I do hope that if you haven't been before, or if it's been a while, that you'll check us out at the website and come to visit. There's a lot of features, and the springtime is a really great time to visit. So that's basically all I'm going to say about Bok Tower Gardens today. What I'm going to talk about instead is just give you an overview of the Rare Plant Conservation Program, because a lot of you probably don't know that Bok Tower Gardens has a Rare Plant Conservation Program, and at least not the details about what we do in the program. So I'll just talk a little bit about why we have a program and what we do, how we go about our work. And then we just finished a three-year project on the demography of the Florida Goldenaster. So you'll be the first ones to get from where I'm going to be sharing that data with you. So, and I'll talk a little bit about the Goldenaster itself. And then finally, I'll just give a slide just to let you know of the current status of the species itself. So first of all, I wanted to mention that plant rarity is a global, really urgent issue. Kew Gardens estimates that about 1 in 5 plant species worldwide are on their way out. They're going extinct, and we all know the reasons for the species decline, and probably all well aware of the implications to ecosystem health as well as our own survival. But there's been not a lot of attention given to plant species loss worldwide. So it's been considered a silent emergency, and it's a really urgent one. Throughout the world, there are these 36 global biodiversity hotspots that are listed, shown here in the colored areas. And these are regions that are defined as having more than 1,500 endemic plant species found just there and nowhere else in the world. But coupled with a big habitat loss of 70% or greater. So there's a lot of species going extinct in these global biodiversity hotspots. And you can see that the entire southeast of the United States, including all of Florida, is considered a global biodiversity hotspot. In fact, Florida has the 3rd most listed plant species in the United States. It's behind only California and Hawaii in the level of endemism and the rates of species extinction. So depending on what site you go to for data, because it can be listed a little bit differently. I believe I got this from the Florida Forest Service website, but it lists 542 species that are found in Florida, plant species that are listed, and 420 of these are listed as endangered. And what that means is that they may go extinct in 20 years or less. So the situation in Florida is an extremely urgent one. I just wanted to show you a map of the rare plant occurrences that are GIS map. These are all the different populations of rare plant occurrences, and I got this from Florida Natural Areas Inventory. And you can see that they're scattered all throughout the state, but in peninsular Florida, they're pretty much centered on these high elevated ridge systems. There's the Mount Dora Ridge, the Lake Wales Ridge has a lot, the Winter Haven Ridge. Over on the East Coast, there's the Atlantic Coastal Ridge. And then over on near the West Coast, there's the Brooksville Ridge, and then many other scattered locations. But a lot of these areas are the elevated, high, well-drained areas that over geologic time had developed these particular habitats that are found nowhere else but their own region. And these plants that are found there have evolved to and adapted to grow only on their particular soil type, in their particular habitat, in their particular ridge system, and are found nowhere else in the world, and they can't really migrate at all. And these are the same areas with higher elevation and well-drained soils that are the optimum for development and roads and groves and railroad tracks and things like that. So as development just increases, more and more of these populations are just getting lost because these plants can't go anywhere else. And there's really no formal entity or no formal agency that's designated to address plant species loss, but botanic gardens have really stepped up to be able to address this. And that makes sense because garden institutions, they already have the expertise and the infrastructure. And because of this, most public gardens worldwide now have an ancillary program that's not part of the visitor experience of the gardens itself necessarily, but works to address regional rare plant issues. And so that's a rare plant conservation program in these gardens. And so Bok Tower Gardens developed one, and it was established in about 1984. So what we do in the Rare Plant Conservation Program is to work under the broad mission to understand and preserve the rare plants of Florida. And we're pretty science-based and research-heavy, and we perform research in the field, or in the greenhouse, or in the molecular laboratory we have on site at the gardens. So in addition to species research projects, we also introduce new populations onto protected lands. We survey rare plant populations so we can update the status for FNA and for state and federal agencies. We restore habitats so they can better support rare plants that already exist on the site. And then we rescue native plants, all native plants, not just rare plants, from impact areas because this genetic material would otherwise be lost if we don't. But one of our main reasons for being is to curate what's called the National Collection. And Bok Tower Gardens is a participating member in what's called the Center for Plant Conservation, or CPC. And this is a national coalition of gardens and other institutions that's dedicated to preserving rare plants of their region. And in Florida, there's a CPC institution that has a national collection in extreme South Florida, and that's Fairchild Tropical Botanic Garden. And because they're located so far south, they hold the national collection for the pine rocklands, the Keys, the Caribbean Basin species in extreme South Florida. And us being more centrally located, we're the nationally recognized repository to preserve the genetic material of rare plants of Central and North Florida. And it's called a national collection because it's not that we're collecting this and it belongs to Bok Tower, we're holding it for the nation, basically. And so what we do is go out to the rare plant populations and primarily collect seeds and preserve those in the national collection. And this serves as a buffer against extinction because there's a lot of material in the collection right now that represents populations that have been extirpated. And anytime we do rare plant rescues, those rescued material, whether it's plants or seeds, those also become accessioned into the national collection to preserve. And so currently we have roughly 3 million seeds and roughly 4,000 growing plants that are grown in these national collection beds at the gardens. For seeds, When we collect them, we store them either at room temperature or at refrigerated or freezer temperature, because the different storage temperatures can make a big difference in long-term viability. And then for a very long-term storage, we also store some cryogenically. We don't have those facilities at the gardens. So the Center for Plant Conservation has an agreement with the CPC to store a rare plant germplasm cryogenically in liquid nitrogen. So and that's a USDA facility. It's NLGRP, and it's in Fort Collins, Colorado. So some of our seeds of Florida are for long-term storage in Colorado also. So in addition to those activities and the curating the national collection, we focus a lot on building and maintaining the rare plant network. And statewide, we do this through organizing with Fairchild what's called the annual Florida Rare Plant Task Force, which you may or may not have heard of. And I'll just mention that it's usually the spring of every year, and we usually have it in person in the venue, but this year it's going to be held virtually. And so there's not too much of a participant limit. And this will be at the end of April. If you're interested, you could look up the more about the meeting and the agenda on the conservation pages of the Bok Tower Gardens website. But this is something that we do to bring the conservation community together, to forge new partnerships, and to share more recent data. I'll be giving a talk there on a different project than Chrysopsis, but yeah, so it's a good meeting if you're interested. Please attend if you'd like, where the registration is open and it's through a Bok Tower Gardens website. And so there's also the Florida Plant Conservation Alliance and the seed collection initiatives, and there's various like federal recovery teams that we're part of as well that are statewide for certain species or habitats. Nationally, I already mentioned the Center for Plant Conservation. There's some Southeast regional plant conservation alliances that we work to build and maintain as well and be part of. And globally, we're part of BGCI, or Botanic Gardens Conservation International. And that way, Florida's rare plant issues get a global perspective as well as a global mention and as well. So we build and maintain networks, which is really important. And then finally, what's really important in all conservation work, so every project we do, everything we do, we try to incorporate education, because conservation is not going to be successful at all in the end unless the conservation education is successful as well. So we do this by involving the public in a lot of our work days through volunteer and public groups that help us, like AmeriCorps or Boy Scouts or Girl Scouts, things like that. We develop projects, student research projects with academic institutions and work with them for the successful implementation of student projects. And this has been from middle school up to PhD dissertation projects. We develop some workshops that we run periodically for education. We develop signage for public parks and public areas, publish and lay articles, and of course there's always the social media that's ever important nowadays. So education's a real part of what we do. So that gives you an idea of the activities that we do to get the rare plant work done. Currently, there's a staff of 3 that are core staff. That includes myself as manager, and I have a conservation biologist, Whitney Costner, and the rare plant curator who oversees the national collection is Anna Clark. And then occasionally, as I get more grant funding, I'll do like part-time or temporary introductory positions, like a rare plant specialist or conservation intern. So those positions can come or go. But right now, there's just 3 staff doing a lot of this work. And because it's a small staff and a lot of work that needs to be done, we rely heavily on volunteers, but also partnerships. We're very partnership heavy, and most of our projects involve at least 1, 2, or 3 real strong partners to get this done. And that's been a real strong component of successful conservation work. And I did want to just mention too that our projects and our program are entirely grant and donation driven. And you know, there's so many things that we want to do, but what we design our grant proposals to add never before found data, like novel data, to help understand the species in a new way, and also to have applied conservation use. You know, it needs to be something that once we get our findings, we could use it on the ground to better support the species. And then a lot of these species are federally listed as well as state. And each of the species have federal recovery goals that are designed by the U.S. Fish and Wildlife. And these are goals that, if they're achieved, the species could either be downlisted or it would be at least way more stable. And so we try to work our projects around meeting those goals as well. So that's how we go about selecting our projects. And so we have a lot of proposals out at any one time, and not all get funded, but we do what's funded. And at any one time, we have about 12 larger grant-focused projects. And then there's always smaller things like, you know, rescues or this or that comes up. So we have a load of them at one time. So we're kept very busy in the program. So, so that just gives you an idea of, you know, why Bok Tower Gardens has a rare plant conservation program, and then how we go about our work. So what I wanted to talk about today is Florida Goldenaster, which is Chrysopsis floridana, population introductions, demographic evaluation, and current status. And the reason demographic evaluation is underlined here is because we just finished up a 3-year demography research project, and the co-authors on that are listed below. It's Scott Ward and Eric Menges of Archibald Biological Station, KT Charton, that's now at the University of Wisconsin, and Phil Gonziska, who used to be my rare plant curator, but he was stolen by Marie Selby to work on bromeliads. So he's now at Marie Selby Botanic Gardens. And this is a project that was funded by grants from the U.S. Fish and Wildlife Service and the state of Florida Department of Agriculture and Consumer Services. So I just wanted to give you a little bit of background on the species, because many of you are probably not interested or not familiar with the Florida Goldenaster. But it's a really gorgeous plant that's only found on the west central Florida. It's a short-lived perennial, and it's pictured here in one of its natural habitats. It's in the Aster family, or Asteraceae, and it's found in scrub habitat or scrub-like habitat in ecotones, and that can include disturbed sites and roadside sites, but it's primarily scrub and scrub-like areas. And it's both federally and state listed as endangered. There's a map on the right of the 5 counties that it's found in. And so it's endemic or found only in 5 counties in southwest central Florida: Hillsborough, Pinellas, Manatee, Hardy, and Highlands. It was federally listed when it was first found in 1986. And at that time, there was only about 9 sites known. But since then, there's been a lot of developments. So some of those sites are no longer exist, but there's also been an aggressive land buying program, and new populations have been discovered, and a lot more surveys were done and new populations were discovered too. So I don't think we'll ever really know the true range, but it's probably stayed somewhere in southwest or west central Florida as well. It would be in this area. And currently there's about 30 populations known. So Bok Tower Gardens has been working on this species since about 2002, a little more, a little less, some years depending on funding. We've primarily been looking at the seed biology, how best to store the seeds, and also how best to propagate the seeds. We do a lot of propagation research and different trials and different variables in the trials to learn how to have the highest germination rate and the highest seedling survival. And so based on that work that we've done since about 2002, we've been able to produce a lot of plants and introduce new populations on the protected lands. And since we've been doing the introductions, we began looking at the life cycle, the natural population dynamics, tracking general population health, and looking to see how well some introductions are doing than others, just using some basic demographic data that we collect every year. And one of the reasons for the recent focus on this species in the last few years is because it's been a candidate now for downlisting. With the 30 populations, about 90% of them are on protected lands, but they seem to be doing really well. But at the same time, we don't know exactly if that's artificial. You can't just go by population size or the number of plants in a population, because things can really change over time. And it doesn't mean it's a healthy population if there's, in one snapshot of time, there's a lot of plants. So that's why we started doing some more demographic work, because this is a candidate for downlisting, federally downlisting from endangered to threatened. Here's some of the data that is just to show you that we've collected over the years on seed quality, germination, and seed storage. And what we find is that when we collect seeds, and we can collect thousands and thousands at a time, we take a subset and look to see whether they're hard and intact and probably viable, or if they're actually empty, because you can't really tell at a glance. So we do this under the microscope. And we find that on average, about 50% of the seeds, only 50% are intact. We also see a lot of variation by the harvest year and by the collection site. This chart on the bottom left, you can see that the vertical rows here or columns here are the different sites that we collected. I know this is small, but just to show you that, like, say from one site, this could range from 17% intact to 63% intact. So there's a big range in all of the sites over years. And then the different color lines are the different years. So you could see that in any given, over the years, the different percent that's intact changes as well. So this just shows the variation in seed quality by year and by site, and it varies quite a lot. When we germinate the seeds in the greenhouse, germination starts about day 6 and goes to about day 35. So you germinate pretty quickly. And we get about an average germination rate in the greenhouse of about 50%, give or take, but that also really widely varies. We've gotten as low as 15%. Actually, we've gotten as low as 0%, but for this, I'm saying 15 to 92. Whoops. And the chart on the bottom right here is just from one site over 3 years, and fresh seeds had about a 68% germination rate. And this shows you too that the storage temperature over time really makes a difference as well. If we just store the seeds in ambient or room temperature, the viability will gradually just drop year after year. However, if we store it at cold storage, whether that's refrigerated or freezer storage, we actually see a bump up, an increase after 3 years in viability. So, so how we store the seeds really makes a difference. And when we do collect seeds, we always do a germination trial for fresh seeds. We'll have a baseline and then pull some out each year from each site. So we'll be able to track it like this. But and then that's in the greenhouse. In the field, germination drastically lowers. It's an average of 50% in the greenhouse, but it could be an average of 0.5%. In the field, and that is reduced even more if there's any leaf litter present. And that's based on data that was collected by Archibald Biological Station. And they also found that when seeds are dropped, you really don't get a lot of germination the first year. And seeds drop about December, January. And so that spring, those freshly dropped seeds really don't do much germination. But the following December, January, February, that's when those seeds start to germinate. So seedlings in the wild are pretty much from seed that was dropped the previous year. So that's some of what we've learned about the seeds and germination rate. And so after learning how to propagate all these seeds into plants, we started doing introductions. And so the first thing we do for introductions is go, okay, we got to find a good site within its historical range on suitable habitat, scrub-like, and also something that's well managed, good well-managed habitat that will be managed well into the future. And there's a few soil types that the natural populations grow on. And so we look to find sites that have those soil series, and that's Archibald, St. Lucie fine, Lakewood fine, Duet fine, and Pomelo fine. So there's a good range of soil types that it grows on naturally. So we look for that. And so we began doing introductions back in the summer of 2008. And at that time, our first introduction was based on 410 seedlings, and we planted these into two sites at a site called, two sites within a site called Cordell East. That's in Manatee County, and it's a FWC owned property. And to date, we have, there's 10 introduced populations to date. There are 4 that were done long ago in 1986, 1987. I don't know, I know one was done by seed. I'm not really sure about the others, and I don't even know exactly who did them, but there's, you know, we do know that there's some record of these being done. But we have done 6 introductions since 2008 that are listed here. And the chart on the bottom left just shows just the increase in plant number at those, at 5 of the sites. Actually, it is all 6, but I was combining Cordell East and West into just one here. And so from the time that they're introduced, there's been a steady increase in plant number. For example, at Cordell East and West, that started out with the 410 plants in 2008 that I mentioned. After 5 years, or by 2015, it was up to 13,000 plants. So there was a big increase there. And so this follow-up monitoring that we do shows that introductions can be effective and they can be successful when we introduce plants. So that was good news. So we did do an introduction at Weedon Island that many of you, I'm sure, are really familiar with. So in the summer of 2009, we met with the Weedon Island folks and chose 3 burn units to introduce the plant. And that's just a map of them here. There's burn unit 2A and 2B, which we just consider one for this project. Then burn unit 3A, and then burn unit 3B. And then in 2010, we began introducing plants, and that we propagated plants into quart pots. And in the summer, and we always do this in the summer to coincide with the start of rainy season, which helps with post-planting irrigation. And into burn unit 2A and 2B, we had seeds that were from 15 different source populations. And so we just track the breakdown, so we kind of have an idea of how genetically diverse the new populations might be, because that can affect how successful the introduction might be, or give us data for later comparisons. So in June 15th, 2010, we introduced 200 plants into burn unit 2A and 2B. The planting team consisted of 16 staff members from the Pinellas County and staff also from Bok Tower, and Fish and Wildlife helped us out as well. And then in later on, a couple months later, we introduced another 722 plants, and those were introduced into burn units 3A and 3B. And so that was that was the introduction at Weedon Island, 922 plants. And then every time we do an introduction, we follow it up by annual fall monitoring. And this is done during flowering season in November. And so this is just to show you some data of how they had been doing at the different burn units. And the burn units themselves were a little bit different from each other when we started too. Burn unit was the most disturbed of the sites. I didn't show any of the soil data here, but it had extremely high calcium levels. And it's also near a trail, and over time we got a little bit of vandalism of the plants and tags, things like that. But from the 200 plants initially put in burn unit 2, after 4 years, you could see the plant number really increased. And the different colors here, the green is the new seedlings, the blue is the number of adult plants that aren't reproductive, and then the red, or reddish color, is the number of plants that are actually reproductive. So right away we started getting reproduction, and then a couple years later we started getting seedlings produced. So they seem to be doing fine there. At burn unit 3A, this is a site that had been roller chopped in preparation for the planting. And this site has done the best with the highest recruitment and survival. The purple here is the initial number of plants put into 3A. You can see how the numbers increased. A lot of new seedlings in the last 3 years where the charts were taken. And then bird unit 3B. This was the most natural maybe of the sites. It was the least disturbed. But so for some reason, the plants have had a harder time gaining a foothold here. The purple first bar is the number of plants put in. And you could see that even though there was a lot of reproduction, there just wasn't a lot of seedlings and new non-reproductive adults being established, and the plant number didn't really go up that much. And a note too is that in 2013, both 3A and 3B were actually burned, and that did seem to stimulate more plants the following year. So burning seems to be good for the species. And this is just a chart of what the overall total population at Weedon Island is during those times, with the initial plants and then the increase over time. So based on just plant number alone, in this time period, it looks like they were doing really well. So from 2008 to 2015, basically our data involved counting all the plants at each of our introduced sites, and we measured the size and recorded whether they're reproductive. And we did find a lot of data from that. We did learn that, there's 4 main life stages in the species. Obviously there's seed, but then it starts as a basal rosette, and it can be 1 or 2 or 3 or more basal rosettes in a plant. And then the vegetative stalks start forming. So the next stage is a basal rosette with vegetative stalks. And then some of the stalks become reproductive. So it's reproductive as a final life stage on this. But what we've done since that time is improve the methodology of our demography. And this will allow for increased accuracy and efficiency in collecting some of this data, because we found that that's something we really needed to do. And the way we're collecting data now is designed to be able to have these different populations modeled in the future, like population viability or trend modeling in the future, maybe some climate change modeling. And it's also better designed to do some analyses to tell us what we really want to know rather than just plant counts. And so it also allows us to identify more of the vital rates that are important to the species. And so we had a funded project for 3 years, from the fall of 2017 to the fall of 2019 is when we collected the data. And through this methodology, we asked, which of the populations are the most or least healthy? How are the introduced populations doing compared to the natural populations? Because that's something that's important for considering whether it needs to be downlisted, or not. And that'll effectively answer the question of whether the population introductions are actually a viable conservation tool in the long run. Do they do as well as natural populations in the long run? And we also looked at what are the effects of microhabitat on whether a plant occupies a certain area or not within a site. And then what is the species response to habitat type? And by this we meant the roadside occurrences versus interior scrub plots, which is something you can't really tell at a glance or from just a plant number. So that's the type of questions we asked. And so what we did was in our study, we did 2 different minor studies within the major study, and one was based on level 3 demography. And what we did here was to choose 10 populations, 10 natural, 5 introduced, and within each population, we set up 10 2-meter rectangular, randomly placed, semi-permanent plots. And in each of those plots, we assigned every plant in there a unique number tag, and collected data on its survival each year, the width of the basal rosette, the height of the tallest stem, the number of reproductive stem, and on the sampling within each quad, we counted the number of actual flowering heads on the plants. And as of 2019, there were 2,880 plants in the data set in just that 1 year. And then we also did level 2 demography in 7 different populations. And this was to study the stage class data. And we chose both natural and introduced populations throughout its range. And this map on the right basically shows where all of our plots are. And so it was basically all throughout the range of introduced and natural populations. So this map includes the demography, level 3 demography and the level 2 demography plots. But in level 2 demography plots, at each site we chose 10 2 meter radius plots, and within each of those plots we assigned each plant to whether it's a juvenile vegetative, which meant it had 2 or fewer rosettes, an adult vegetative, which was 3 or more rosettes, or whether it was reproductive and had 1 or more reproductive stems. And the number of rosettes in that case was irrelevant. And so we counted the number of plants in each plot in each stage class. And as of 2019, we had over 3,000 plants in the data set. And what we also did was to collect microhabitat data in both randomly placed occupied and unoccupied plots. And occupied means that when we chose random plots, if it had Chrysopsis in it, it was occupied by Chrysopsis. And then areas that were just random plots within sites that had no Chrysopsis. So those are the unoccupied plots. And we chose both populations that were both part of our demography and stage class populations as well. But that was what we did, collect microhabitat data, and that was only just over 2 years. And so let me just get into the results to show you now. And all the results, all the analyses, by the way, was done by Scott Ward at Archibald Biological Station. So he actually gave me these charts as part of it. So what he had done is do like just a basic t-test to look at the averages on some of the vital rates. And this is showing the chart on the right is showing the t-test done on annual survival, which is AS, and on seedling annual survival, which is called SAS. So annual survival is of all the plants, and then seedlings is just the survival from year to year of just the new seedlings, which is a second important vital rate. And on the charts on the right, there's AS, the annual survival from the years 2017 to 18 and from 2018 to 2019. And the bars, the black bar is the introduced, and the lighter colored bar is the natural populations, called the wild populations here. But in both of these years, year to years, the annual survival was statistically higher for introduced over natural populations. And then the same was seen for the seedling annual survival for both years, that there was higher annual survival for the seedlings in introduced versus the natural populations, and it was statistically significant. And then we also looked at the scrub habitat versus roadside, and the scrub was more like a habitat interior, the interior scrub, rather than the fire breaks or anything that was more disturbed. And so that's the chart on the bottom right here, and that's the annual survival over those 2 years was statistically higher in the scrub areas than the roadside. And then same with the seedling annual survival over both of those years was also statistically higher with the scrub habitat rather than the roadside. And so also we found in introduced versus natural and scrub versus roadside, and I didn't show the data here, but there was more flowering heads and higher seedling recruitment in the introduced populations. And same with the scrub, that there's more flowering heads and higher recruitment in scrub versus roadside. So that was something that would be important to say, that introduced and scrub are have healthier populations. And so all of the data that we collected, Scott put together to standardize across the populations and score all the vital rate results in terms of population health. And so he did this by Z-scores, and the Z-scores basically are ranking, taking all the data and ranking them by population health. And the mean here on the chart on the right, the 0, that's like just the average health. And so anything above that is a healthier population than average, and anything below that is a lower population. And this is ranked by healthiest to least healthy. And this shows that a couple of the introduced populations in the black here are the healthiest populations. But then there's also some natural populations that are healthier than average. And then the same with lower than average health. There's both natural and introduced populations that are among the least healthiest. And also those on scrub versus disturbed or roadside, those on scrub habitat were also the healthiest. And so overall, what this is showing is that it gives us an ability to use all of our data to rank which populations are the healthiest. And it also shows us that overall, the introduced populations seem to be healthier than the natural populations, which was interesting. And so going to the level 2 demography results, which was looking at just the stage class. And this is another way to really look at population health and not just look at numbers of plants. But what you want to look at here for stage class is that the best proportion, the healthiest populations, what they have is a good number of small vegetative plants as well. And that means that a lot of the seedlings were produced from last year and survived. And so even if you have a lot of flowering, which is the darker gray here, if it doesn't translate to a good number of small vegetative following year, then that is not necessarily meaning a healthy population. And so what we find here is that the healthiest populations on average for over the course of the 3 years was the 4 here on the left. And two of the 4 are introduced populations. And so the least healthy were the 3 over here on the right. And these had consistently low counts and lower proportions of vegetative to flowering. So these were considered the least healthy. And we also looked at microhabitat of the occupied and unoccupied. And what you can see here is that the occupied is a dark bar, and the unoccupied, meaning there were no Chrysopsis in that area, is a lighter bar. And what was statistically significant here was that the occupied sites, where Chrysopsis could grow, had statistically more bare sand cover and a diversity of forbs in the understory. And then the unoccupied sites where Chrysopsis didn't grow had statistically more leaf litter, woody shrubs, and more of a subcanopy. So that shows us that the presence of these plants negatively correlates with litter, shrub, and subcanopy. And then down here is just on the left is just a photograph of what good habitat looks like for the Chrysopsis, one of the introduced populations. So here's a summary of what we found from those 3-year study of the demography is that introductions do seem to be a good conservation tool for the species. They did a little bit better than the natural populations. And also this, having it actually on scrub habitat and not just roadside or disturbed seemed to make for a much healthier population as well. And it seems like population health seems to be a factor of the habitat conditions rather than just say soil type or plant number, something like that. But it definitely needs a good habitat. And then of course, plant presence, is not going to be supported if there's going to be a lot of leaf litter, oak, woody shrubs, or subcanopy. What you need is bare sand and a diversity of understory forbs to help support the species. And then finally, what I wanted to talk about the Chrysopsis is something that we helped with but weren't involved with. This is from the U.S. Fish and Wildlife Service. They do species status assessments of the federally listed species and collect data from all the different land managers, anybody who's working on the species, just to try to find the most and do an analysis of the most up-to-date data. And the most recent one they did was in 2018. And so this is from the 2018 SSA for Chrysopsis. And what they did was to rank how the populations or project how the species as a whole is doing, is to look at it in terms of its resilience, the resilience of the different populations. And this is based on 3 factors they looked at: the population size, the level of habitat protection, and the habitat area. And what they found was that there was 7 very highly resilient populations, 11 highly resilient, 6 moderately resilient, and then 6 populations with a low resilience. And that's out of the 30 populations currently. And so then they looked under 3 future scenarios. One is like if status quo, if nothing changes, no introductions are done, no management, additional management, nothing else is done. And then a pessimistic one where absolutely nothing is done at all, not even just the baseline management. And then the targeted conservation, which is if efforts could be put into conservation of the species, what it would have. And what they found is that under all 3 scenarios, for the 7 populations, the ones that currently have a low resilience, those actually become extirpated even under the best case scenario. Under status quo scenario, the number of highly and very highly resilient populations drop from 18 to 12. Under the pessimistic scenario, 3 more extirpated populations disappear. But under the scenario in which conservation efforts are put towards a species, the number of highly and very highly resilient populations can increase to 27. And so this shows that more work will be needed to help preserve the species. And so currently, just to let you know, they estimate that there's probably about 50,000 plants in the wild, 50,000 individuals. And 12 of these populations have 1,000 plants or more, and the rest have less. But 90% of all the populations are on protected land. But what I found interesting too is that half of all the plants, half of all the 50,000 individuals are at introduced populations at 8 sites. So this shows also that introductions are really important to help prevent extinction of the species. Where we would like to go with the Chrysopsis work is to work to see if seed sowing is a viable way to introduce healthy populations, because it does take a lot of work to introduce plants into the wild. It takes a lot of manpower, a lot of greenhouse time and space, a lot of the plant propagation and ferrying the plants out there, a lot of work planting it, and then the post-planting irrigation needs to be done every other day for about 3 weeks if it doesn't rain. So it takes a lot of work and resources to do that. And so what we looked at this year is to do a study at 4 different sites throughout its range, and we introduced seeds from 2 sites in all of these 4 populations in January, and we're going to do it again identically in June, and that will compare seed introduction two different times a year. And so we may be able to find out if there's a, seeds work just as well for to introduce new populations. Because I know that one of the 1986 and 87 introductions, the one down at Fort DeSoto, that was actually done with seeds. And so we're trying to revisit that again. So that's something we'd like to continue working on. We're going to continue doing fall monitoring of the introduced populations, but it would also be nice to continue some of the demographic data that we have been doing. So for future population viability analysis and modeling, population modeling, more in the future. It'd be interesting to look at the soil microbiome, because that could be some important feature of whether a plant persists or whether the populations can be healthy. So it'd be good to look at the microbiome of some of these populations. and to correlate health of the populations with management histories like fire, and then maybe correlate some of the population dynamics with rainfall or other climate change parameters. So that's where we hope to go with it in the years in the future. And so I just wanted to do some shout out acknowledgements to folks at Archibald Biological Station that helped with our 2-year study. That's Scott Ward, KT Charton, Eric Menges, Annika Rose Person, Stacy Smith and Stephanie Coons. There's Bok Tower Gardens and staff, including former, that's Phil Goncisco, Whitney Costner, Cindy Campbell, Ann Clark, and Joshua Liu. Mike Ball Naturalist helped very much with these projects over the years, and Todd Mecklenburg of Fish and Wildlife, Mike Jenkins of Florida Forest Service, and to thank all of the landowners of these populations who helped support some of this work. And so with that, I just want to thank you for your interest and your attention, and I'd be very willing to answer any of your questions.